So today, I have a very special guest from across the pond in the UK named Mark Lintern. And I'm just really happy that you're here. And I'm excited about the conversation we're about to have. So welcome.
Thank you, Casey. It's wonderful to be on your podcast. I really appreciate it.
Would you do me a favor, man, and just like give me an introduction to our audience if they don't already know you, like how you ended up being in this position?
Sure. I'll try and do the really short version. So, um, my background is actually in graphic design. So, I'm not a clinician. I'm not medically trained. However, I've spent 8 years researching the medical literature after I developed cancer myself. Um, I wanted to basically determine what DNA genes were the cause of my particular cancer, and that led on from one thing to another. I discovered different theories that were available, and I realized through that process that the underlying mechanism behind cancer was still up for debate. The metabolic theory came out for me as on top, and I carried on my research because a friend of mine developed cancer, and I wanted to help her with all the research I'd already done. She unfortunately passed away. Um, but as I went through the process, as I was learning more and more after the years, and it became a passion, really, an obsession, some would say, I began to notice there were one or two contentions with the metabolic theory that didn't sit too well with me, that needed, not saying that the metabolic theory is necessarily incorrect, it just I needed those things to be addressed and still need to be addressed, I think. And that led me on to more research, but I realized that the Warburg effect was key, or abnormal metabolism, as we see in all cancers. Um, to cut a long story short, 8 years later, I identified a correlation that suggests a different mechanism or an alternative mechanism can explain pretty much most of the hallmarks of cancer as well. Um, and I developed a new theory called the cell suppression theory after 8 years of meticulous research of the peer-reviewed literature. From there, because of my graphic design status, my citizen scientist status, I needed credibility. So I contacted a cancer charity. And with that cancer charity in the UK, we organised a big event where I debated 10 experts on cancer, four scientists, three clinicians, active clinicians, and three patient experts in a panel over 6 hours. I debated them, presented my research in front of an audience, a live audience of 200 medical professionals, and everyone voted. The panel voted, the audience voted, and the vote that came out was 7.4 out of 10 for the panel, and overall with the audience it was 7.9 out of 10 in acceptance that I'd generated a significant medical, not breakthrough, but a theory that is worthy, or a hypothesis that is worthy of investigation. And since then, I've been working with a number of scientists each month, sorry, we meet, they volunteer their time to establish um, a pilot study to test the theory. And very recently, from November last year, I've got a biotech company on board who now want to fund this and take the study forward to initially test the theory. So I've got a lot of things going on from the scientific perspective. I published a book 2 years ago, written for the layperson to understand my theory and how it fits in with all the other theories, um, and how the mechanism I identify relates to cancer and how it can possibly improve or be an addition, offer additional treatment options for cancer patients to work with whatever protocol they're currently undertaking. So it's been a big undertaking. It's about 10 and a half years since I started the whole process. Um, and this is where we are now.
That's awesome, man. I mean, that is a very touching story, man. I think especially the fact that like you're motivated not only by yourself, when this journey started, but when your friend, developed cancer. And, I can't help but say that I felt touched, unfortunately, when you did the work, or at least at that point, some of the work, and unfortunately we're not, you know, y the warnings that you were giving were not heeded, unfortunately. And, unfortunately, I think I'm currently doing a course right now in the evenings this week, and I was kind of telling people how, it's cheesy as it seems, um, finding people who are supportive of your decision to not necessarily only go with the standard of care, not saying not going to do the standard of care at all, right? Um, is critical because, if I had a nickel for every time I heard someone say, "Oh, my family said, oh, just listen to your oncologist," they went to school for 25 years, they know what they're talking about, and it's mostly out of, I think, fear, but also love that people say that to people, and it really makes it hard to get through to people sometimes, even if you're right, even if you have really good information. And it really kind of made me sad to hear you kind of like basically lay that out with about your friend. And I think that it's just, if there's nothing else that we can agree on today, is that I think that, by watching several podcasts with you, in the last couple of days, because I wanted to understand you better and understand what you're trying to, bring to the scientific community. I think you want to help people, I think you want to um, overcome what has become entrenched dogma. And, as much as you may have to deal with credibility, unfortunately, because you don't have a PhD behind your last name or whatever else title. Um, I think that you have a lot of advantage in some ways because you haven't been indoctrinated. I mean, not to the degree that like a lot of us have, right? Like some people have their entire career based on the somatic theory. Like imagine that. And so I think it allows you to be more creative and more thinking outside the box without being constrained. So I do think that in a lot of ways is a huge advantage. I also wanted to, before we get started, because I do have, I'll be very frank with you. Like I have I've listened to your podcasts and I've read your theory, at least the free version on the internet, on your website, which I think is beautiful, by the way. Like I said, the graphics
Thank
that
you.
you did are unbelievable good. Because the graphics that I put in my videos are like from research papers. There's no Casey Peavler making graphic design stuff. So I do think it's impressive. But I think what's impressive more than anything else is the fact that you do have a background in graphic design and you have come from there to become effectively a biologist, a cell biologist, like a biochemist. You have a strong, background, obviously, in immunology. I've heard you speak, and for hours now at this point. And I just think it's impressive the ability, to synthesize that much information and then go and be able to, speak to a group of panel of doctors and or clinicians and or researchers and have 6 hours of debate and actually have that kind of result. I think that's pretty unbelievable. So I just want to like give you that. Um,
Thank you.
so I guess maybe the first place I'd like to start is for you to give a um, a basic overview of the cell suppression theory and maybe perhaps even the evolution of, going from I want to know the mutations I have in my cancer and kind of being disillusioned by the answer you got from your oncologist and maybe even the medical literature and how you evolved and how it's even evolved maybe since the book came out 2 years ago. Like, what have you learned that has maybe changed you a little bit even from then?
Okay, well, thank you for all that. I resonate with everything you were saying there. And I could go off on a load of different tangents and talk about all those things because it's quite profound. And really, what I'm offering with my theory is I'm not saying I'm correct. I'm just saying I've tried to, and I'm not backed by anyone. All the 8 years of research I've done is at my own expense. So, it's an individual desire to essentially find truth because I don't want to go through what my friend went through a long time ago when she passed away. So the idea was to help protect my family and friends. So this is where it comes from. This desire to identify truth and to help as many people as possible. Um, so with that in mind, like you say, I don't have any prejudices or I was able to be objective. And, I was, and I still am on the side of the metabolic theory. I think it's profound and it features heavily in all cancers. Um, but I'll go through the whole process of how I basically got into it and how that developed and changed. So as I went through the process of having my own diagnosis, I did question the oncologist and my GP to ask DNA mutations caused it because that's the dogma, as you say. And most people are taught that. Medical professionals are taught that. That's the main premise in the medical textbooks. Um, and they couldn't tell me. So I thought, well, I really want to just research this myself to just get the lay of the land and try and work out how I can prevent this from occurring in future for myself. And as I went down that rabbit hole of trying to establish a pattern of DNA mutations, which is what the somatic mutation theory is suggesting, a sequential pattern of DNA mutations is likely the driver of cell proliferation, unbridled cell proliferation. It became clear that there weren't any obvious answers, and it was a lot more complex and confusing than the layperson is necessarily led to believe. So one of the main things was the fact that the mutations are found to be random, even within the same tumor. The same cells that are allegedly generating the disease, the random mutations. There's also the contention that there's not enough mutations in many of those. On average, there's about 1.7 mutations have been found recently in a number of studies. 1.7 mutations per cell. And arguably, there needs to be a few more than that to drive the hallmarks of cancer. And then there's other tissue where tumors have developed without driving mutations, tissue where you have healthy tissue with the alleged driving mutations that cause the disease, then the disease doesn't develop. Then you have the DNA transfer experiments, where they try to transplant basically uh, mutated DNA from tumor tissue into healthy tissue, healthy cells, and expected tumor growth to develop.
They
Unbridled
transplanted the
growth.
entire nucleus, right? Like the entire nucleus, the
Yes.
entire genome. It wasn't just the bad parts, it was the entire cancer nuclear genome.
Yeah, so exactly. If that's signaling unbridled growth and that is the underlying origin of the disease, then we should have that in the cells that it's transplanted
Makes sense
into,
to
and
me,
we don't get that. We don't
right?
get that. So, um, and then Professor Paul Davies, who is the co-author of the atavistic theory, when he came out and published his in 2013, he talked about National Cancer Institute doing a deep dive into the medical literature and found that concluded at least 80% of the foundational studies that form the backbone of our understanding of cancer, i.e., our studies into the somatic mutation theory and the genetics of cancer, up to 80% of them can't be replicated, which suggests that the foundational basis of what we think cancer is based on the somatic mutation theory is predominantly incorrect or flawed. Um, so that's also a big red flag saying, okay, something else must be going on here. And then when you start looking outside of that paradigm, and I stumbled across the number of other theories after I started questioning the somatic mutation theory, then this huge area of research opens up, and you realize that you were just looking at a really small area, and that when you listen to Thomas Seyfried and you listen to proponents of the tissue organization field theory, you realize that actually the origin of cancer isn't set in stone. The science isn't settled as
The
we're
science is never
we're
settled.
well, exactly. The science, it's an ongoing evaluation of new data, isn't it? Forming new conclusions.
Right.
Um, so, I mean, I looked at everything from the viral theory, pleomorphic theory, atavistic theory, tissue organizing field theory, cancer stem cell theory. And it's only when really coming across the metabolic theory did that, you know, things start to fall in place. I mean, for me, the cancer stem cell theory and the tissue organization theory are also paramount because we're talking about tissue inflammation. And inflammation is one of the hallmarks or one of the guiding principles of the development of cancer because from inflammation, you get most disease developing. It's a natural response to any sort of cell damage or danger. So that's a major hallmark, and that theory really delves into that process of how inflammation can damage the ability of tissue to communicate uh, within that tissue, and that could reduce the ability of the constraints, the signaling constraints to stop cell proliferation. So
Mhm.
it's key to look at inflammation. So that theory is paramount. But also, we're aware that cancer stem cells exist within tumors, approximately about 2% from the research that I've done. And they play a huge part in chemotherapy resistance and regenerating the tumor itself, even driving the tumor. So that needs to be taken into account. And I don't think,
Do you know what my epiphany was, Mark, with the cancer
go
stem
for it.
cells? Because the vast majority of traditional chemotherapy and really oncology agents is based on the fact, and this is kind of what I learned when I was in medical school. And again, I'm not an oncologist, so therefore maybe I would have learned something different if I went to in a fellowship in oncology after my internal medicine residency. But suffice to say, basically what we're taught is that these cancer cells are clones of themselves
Mm,
and they're continually growing, right? So because they're continually growing, our treatments are based on will stop cell replication, alkylating agents, put in like fake nucleotides,
mm,
block microtubule formation, block the cell cycle. And what surprised me about the stem cells, and part of the new hallmarks of cancer that came out in 2022 was the idea that these stem cells can be senescent. They cannot be
mm.
growing, right? And so therefore, everything we have in our armamentarium from conventional oncology would be not effective because those aren't going through the cell cycle and replicating all the time. And I think that was, I mean, other than the fact what you're talking about with, driver mutations not being part of cancers or healthy tissues with driver mutations, or, I mean, there was a podcast I watched with Jason Fung. It was Jason Fung. It was Thomas Seyfried and another oncologist. I can never remember his name, but he was the host. And Jason Fung was talking about how, and I didn't know this at the time, that in one tumor there could be like 10,000 different mutations. Like, how
Mm.
can you have targeted therapy with 10,000 different targets? I mean, it's biologic whack-a-mole, and it's absurd. And so basically, you came to the same conclusion that the current paradigm, at least with the evidence we have and non-rep being able to be replicated, is absurd. And I'm being treated, and my loved ones are being treated based off of this theory, And it sounds like you were extremely yeah, systematic with looking at, okay, this is the other theories. I think that's really cool. And then, and then what happened? Like, how did you synthesize? How did you go from there, make the leap to like, wait a minute, I have my own theory.
Yeah, so really it's all about for me the consistency. So the randomness of the DNA mutations couldn't explain the consistency of cancer, and the consistency is the hallmarks of cancer. Regardless of what tissue it is in, generally you have the same disease occurring. I would say, well, that at least you have this consistency. So the metabolic theory highlighted there was explaining this consistency through this switch in the Warburg effect or abnormal metabolism. And that really resonated because that explains a hell of a lot of what's going on. And when you have a reliance on glycolysis and the production of lactic acid, that explains a number of other things, such as angiogenesis, because excess lactic acid generates angiogenesis. But there was always this confusion in the primary medical literature saying, well, they were still struggling to identify the DNA gene or mutation that would generate this angiogenesis. We all know the pathway, and that's upregulated, but the why is the important thing. And a large amount of lactic acid being present is one of those drivers of that. So I realized that consistency was key, that the Warburg effect was also key. Um, and then from there, I carried on in the medical literature, looking at other things, and I was doing research, not even associated with cancer. So I was looking at infection. I was just generally
Hmm.
interested about how the human body works.
Hmm.
And because I had this consistency in mind and the Warburg effect forefront of my mind as a key feature, I stumbled across a paper which described how the infectious process occurs. And when you have an infectious threat, cells, not just immune cells, but epithelial cells and other cells of the body, they respond through a cell danger response, which triggers a metabolic shift to a Warburg-like metabolism. That was the phrasing that was used in the paper. And that was the light bulb moment for me at that moment in time was, okay, we've got the Warburg metabolism occurring in cancer. We've got Warburg metabolism occurring in response to an infection. Hmm. I then wondered if they could be related. And then I did a lot more research to try and find out if this had already been discussed in the medical literature in relation to cancer. But all I could find in terms of infection causing cancer of which 20% are attributed to viruses and one or two bacteria, it would be the viruses and the bacteria would cause damage to the DNA, um, and that would then transpire into the cause of cancer. So it's not that the pathogen themselves is orchestrating anything in particular, it's causing the damage that then leads on to the disease. But then I got to think, well, okay, does this literature suggest what if the pathogen remains? What happens to cells that are infected? Um, if the pathogen remains, the literature pointed out that the Warburg effect remains because it's a cell, it's an anti-infection response. And then I came across Dr. Robert Navio's work.
Hmm.
Now he describes the cell danger response model he puts forward. Um, and that's very interesting because I'll discuss later on. He points out exactly what's going on in the cell. But he highlights again this metabolic shift that occurs because of the infection. So then the question became, well, could it actually be that tumors are responding metabolically in the way that they are that we're seeing? It's not because of damage to the cell. It's because the cell is being suppressed somehow, being hijacked by the pathogen. Could the pathogen still be present? And that's driving the Warburg effect. And the medical literature does confirm that that is a possibility. Then I discovered evidence from Ravid Straussman, Rob Knight and colleagues, where they've actually studied the microbial signatures within tumors and cancers, and they found that bacteria and fungal pathogens are present in all of them. It's now known as the tumor-associated microbiome. So now we have this model in my head where there's a potential shift in the paradigm of what cancer is, that we have these pathogens present. We don't quite know yet what they're doing because this is a new field of study. Yet I've already created this theory which suggests that actually they're driving the Warburg metabolism that we see, because that's the shift that occurs when cells are defending themselves against infection. And then we have them present in the tumor. So they're there, they can orchestrate this shift. And then as I've gone through the process of thinking, well, okay, could this be a new mechanism? Um, can I explain the rest of the hallmarks? Because that's the metric by which cancer theories are measured, the validity or the
Hmm.
accuracy of cancer theories.
Well,
I
that's the
mean,
way they should be, right? Because if that was the case, then the somatic theory would have been debunked. Because you said multiple times I can only kind of explain about two of them. So you would think that
absolutely.
would be the case
Yeah.
with that.
I spoke to Thomas Efrey, like I've been in contact with him quite a number
That's
of
what I heard.
times in the past whilst I was doing my research. And he was the one who kind of suggested, really try and emphasize looking at these hallmarks, explain your theory through this mechanism, because this is one of the key mechanisms to explain and give validity to your theory. So I then went through the whole process. Can I explain all these hallmarks through the interaction of these pathogens, specifically fungal pathogens, um, on cells and with the immune system as a whole as a systemic organism? And yes, the literature is abundant in the ability for me to explain all these hallmarks. But not only that, as I was going through the literature, I realized there were a number of other features of cancer that weren't fully explained enough for me by the other theories, such as why is iron a double-edged sword? It's required for our cells for healthy function, but then it's also in the process of being considered a carcinogen because it can stimulate the disease. So there's another 20 of these that I thought, well, if I want to have credibility, if my theory wants to have any sort of substance, I need to explain all these as well. And I've done that along with a number of other ones on top of that. Um, so that whole process for doing that, explaining the 10 hallmarks in a comprehensive fashion and these are the 20 features, has kind of given my theory now credence, because it's highlighting that I can, through this process, through this simple mechanism, I can actually explain a lot of the missing parts of the puzzle, if you like, and show how all the different theories link together. And the one thing I would say that I think I brought to the table is a slightly different way or a completely different paradigm, in the sense that when I looked at all the other theories, metabolic theory included, they seem to, I think, start from a particular flaw in thinking. Um, and I've always tried to approach this from first principles thinking. And that is that the disease is thought to be initially or assumed to be a malfunction-based disease.
Correct.
So,
You have a great graphic in
re-
here, by the way, on this.
yeah, thank you. And so, what you've really got is you've really got only one umbrella theory, which is the malfunction theory of cancer. And then you have all the other theories, semantic mutation theory, metabolic theory. All those other theories are sub-theories of that notion that cancer arises from cell malfunction. That is, part of the cell no longer functions correctly, it's damaged. That then leads to unbridled proliferation. The cell develops a mind of its own. It's gained autonomy essentially. It's lost its the control mechanism that's keeping it uh, in the state of quiescence um, as it currently is. So now it's developed a mind of its own. It can do what it wants. But what I'm saying is actually there's a completely different paradigm because we're viewing things through this cell malfunction paradigm. We're not stepping outside the box and then looking at other possible paradigms. And the paradigm I'm putting forward is that actually potentially cancer is a suppression-based disease, and that opens up the door to external forces such as intracellular as pathogens uh, hijacking cellular pathways and manipulating or modulating the immune system um, to further its goal of survival within the cell. And then that leads to the proliferative state we see and all the other hallmarks that we find occurring. So it's this complete shift. It's not just another theory. It's a cancer isn't a malfunction. It's a potential suppression-based disease.
And very fascinating. We'll highlight that I'm a picture person, and you have a great graphic, like I said, on your website that actually gives you this kind of like compare and contrast, and I think it's excellent. What I'll say is that I actually think that both can be right at the same time,
Mm.
and I'll tell you why. Because I've been studying you for the last several days and reading your website, I have went to Naviau's, paper on the death response or cell danger response that went to, I think it was initially published in 2015, and then he had another one that was published in 2019. I read both of those papers, and I thought it was extremely fascinating. I like to learn, and I like mitochondria, so it's heavily based on that. And so, um, I noticed a couple of things. Okay. So, for example, the cell danger response, where the infection would be present, active inflammation, active immunity would be cell death or cell danger response phase number one.
Mhm.
And in that phase, he talks about within minutes, the cell is able to change from oxphos to, he says at least in the table, and I could not get more information about it than what he had. And I tried to go to other papers trying to find, you looking at references, etc. But he says there's glycolysis going on during that time, and that oxphos is effectively curtailed. But he didn't say anaerobic glycolysis. He didn't say aerobic glycolysis, and it kind of bothered me because the reason why is because in phase two, in or cell danger response phase two, it specifically says aerobic glycolysis, aka the Warburg effect, right?
Yeah.
And that, at least in his paper, I like to learn more, was only happening within the stem cells and the immediate daughter cells in the area for rapid proliferation and growth. And where I saw the crossover happening was the fact that, let's say hypothetically, it really doesn't matter if it's anaerobic or aerobic glycolysis that's happening in phase one where the infection is still present. That is driven, and you'll see it in his phase two. He talks about this. It's basically the loss of the chaperone proteins for an important critical thing within cancer, across cancer. Okay, like it's talked about everywhere, and that's called the hypoxia inducible factors. And the hypoxia inducible factors are what's responsible for, at the cellular level, the communication to change, from oxphos into the Warburg effect, and it'll enhance glutaminolysis as well. And so my point is, I was like, okay, there's clearly like what Mark's talking about, and what, I probably butcher his name, Naviau, is talking about. There's a common mechanism here. And what I came to the conclusion is that, let's say hypothetically, in an acute infection, this is a pretty rapid process.
Mm.
What we know, the reason why H. pylori will increase your risk for maltomas or the fact that you have chronic viral infection causes cancer, at least what we believe, it is the chronic inflammation that happens there, right? And the chronic inflammation has collateral damage because there's a lot of reactive oxygen species, a lot of signaling going on, et cetera. And so my point is that if HIF1, let's say hypothetically there's a chronic fungal infection in this case, you would have to imagine there's chronic inflammation, chronic oxidative stress, both of which can signal through HIF1 alpha. HIF1 alpha um, will do the metabolic reprogramming you're talking about in the acute phase. And it would, if it was chronically stimulated, I agree. I think it could totally explain what you're talking about. But what I would tell you is that one of the things that is inconvenient about HIF1 alpha is that it will cause pathologic excess mitophagy. And mitophagy um, is the removing of mitochondria, removing of mitochondrial mass. So my thought is that this is me just like spitballing with a friend from the UK. Um, is that ultimately it would end up where there are less mitochondria number, potentially damaged structure and function because of the chronic inflammation and infection that's going on. And you're ultimately going to lead to a Thomas Seyfried, basically mitochondrial dysfunction type of situation at some point in the chronic disease process because HIF1 and inflammation are chronically being, driven. And unfortunately, HIF1, and this is where I think I'm a little bit different than you. It can be stimulated by many things. Circadian dysregulation, chronic oxidative stress, chronic inflammation from any source, right?
Mm.
Deficiencies. So my point is that pseudohypoxia, as it's called, because it can be activated by hypoxia, as you would imagine, hypoxia inducible factor, by low oxygen levels. I'm not living at the top of Everest. I don't have obstructive sleep apnea. I don't have an end-stage COPD where I'm wearing oxygen. So where would my hypoxia come from? Well, there's an entire area called pseudohypoxia. And pseudohypoxia can be stimulated by NF-kappa B and other inflammatory cytokines. And that's where I think the connection could be between your theory, for sure, and this theory, no question. But I think that they could potentially both be true. Maybe the beginning, maybe, there isn't significant mitochondrial damage. It's just the actual acute, cell danger response that's leading to the metabolic reprogramming. But chronically, maybe that could lead to a Seyfried like type situation. I don't know what you thought about that. I just wanted to share that with you.
Yeah, no, thank you. Well, this is what I love about speaking to people like you. I mean, you're so in depth about all the pathways involved, and I can tell you're the same. You're similar in the sense that you really want to get to the truth, and you're not afraid to ask difficult questions. Um, so for me, with mitophagy, that would be the cell when it's getting damaged. It's attempting to eliminate the damaged mitochondria in order to make the cell more efficient, and mitochondria control of the cell more efficient. But yeah, I completely agree. Over the course of the development of cancer, you're going to get, and with the infection going on, you're going to get, and with the reactive oxygen species, progressive damage to mitochondria. So you are going to see what Professor Seyfried points out, this drop in the ability of mitochondria to function effectively, which could explain the shift over to aerobic glycolysis. Um, so I agree. I'm not saying that the metabolic theory is wrong. Um, but for me, how I explain the, the, the functioning of the cell danger response model, and the reason why fungi, I think, are intrinsically involved, um, is because how Dr. Robert Navio explains the process is that mitochondria are a lot more than just the energy creating factories in the cell. They pretty much mediate a lot of the cell functionality, don't they?
Totally agree.
Um, Especially defense against pathogens as well. So the process being that, I mean, we still have oxygen being absorbed into the cell in areas of the tumor that aren't hypoxic. And
I've heard you mention this before. I don't believe the oxygen is necessarily absorbed, right? It's just diffusing across a
diffusing.
gradient, right? It's being
Yeah.
used. right? It's being consumed, right? And that's where I think that that's where I mean that's where medicine, and PhD researchers get it wrong. In fact, there's a whole section from Seyfried's new paper talking about how oxygen consumption rate does not equal oxphos. It's very clear
I
that's not the
yes,
case. It's very
yeah, I
ambiguous.
agree.
And you're talking about it being consumed by ROS to fight off the pathogen. I've heard you say this before, but I just wanted to
Yeah.
put that in there.
Yeah, sorry. Probably coming from my graphic design background and not being talking to scientists on a daily basis all the time, I'm going to say the wrong type of word every now and again. So, diffuse should have been. I should have used that instead of absorb. Thanks for pointing that
No
out.
problem.
Um, so, I've lost my train of thought just
Sorry,
slightly.
man. You were talking about how the oxygen is not as present for oxphos, I think, because I've heard you talk about this before, how oxphos is suppressed and how the oxygen is being used to generate reactive oxygen species to fight off basically to fight off the pathogen.
Yeah, so the notion is that the mitochondria actually this is intentional. So this switch to the Warburg effect is intentional. We see this in immune cells as well. So mitochondria repurpose the oxygen they would normally use to generate ATP um, to utilize that and the electrons to produce reactive oxygen species in order to defend the cell against the pathogen. That then means that there's a reduced capacity of mitochondria to generate ATP, so it needs it from another source, either glutamine fermentation or glycolysis, which is why we see the Warburg effect occurring. Um, the issue I have with mitochondria being defective, and that's the reason for the switch, is because in a number of studies, it shows that OXPHOS can be upregulated. And I know there's contention with that, and I agree with Professor Seyfried and yourself, what you just mentioned, is in the sense that when a number of scientists are measuring the oxygen consumption rate, that's not reflective of actually
Correct.
mitochondria producing ATP. It could be glutamine fermentation occurring, and that could be the energy ATP being generated through that process. And so I agree with that. But there's a number of studies that show that if you use inhibitors of certain complexes, um, I think oligomyosin from
Hmm.
memory inhibits complex 5. What we see is a drop in ATP and then a subsequent increase over a 4-hour period of lactic acid output to compensate for the drop in ATP, suggesting that it's coming from OXPHOS. Um, Also, there's studies showing again that OXPHOS is upregulated when you have nutrient deficiencies of different kinds occurring. Um, and specifically, there's another issue with prostate cancer is quite a big black swan, I would say, because initially in prostate cancer, the Warburg effects doesn't occur. So we have OXPHOS, which is generating prostate tumors, essentially. And that's a big deal because that's one of the biggest, well, it's the most dominant cancer in men. So that's suggesting that defective mitochondria don't necessarily well, they're not defective necessarily you haven't got OXPHOS deficiency occurring in prostate cancer, yet you have prostate cancer developing despite that.
I want to push back a little bit on
Right.
that. So there was a paper published in February of 2025 that kind of debunked that thought, actually. It was interesting. It was a paper that was talking about CLL and prostate cancer as being these thought to be these different entities. And it basically talked about these are fat utilizing cancers. And I'll be honest with you, man. There's several papers out there that suggest that many cancers are utilizing quote unquote fat. And when you actually read the paper, like when you actually get into the nitty gritty of it, they're not using it to burn energy for oxphos. They're using it to make other fatty acids because, membranes are at least 50%, depending on the membrane. And because there's so much cell replication, that's why they need the raw materials. So, and that's also why, citrate is made by cells because citrate is the first step in de novo fatty acid synthesis. So when you actually look at the metabolomic studies they do, they'll make suppositions like there's going to be oxidation of these fatty acids, But this paper in particular, which has nothing to do with Seafried, basically said, well, I think that the predominant paradigm that these cancer cells are using fat for fuel is likely wrong. And I think that that's backed up a lot. I mean, if you haven't read the paper from Dr. Seafried's group in April 2025, I highly encourage you. It will take all the sacred cows of the conventional, oncology and other kind of like thoughts. And it just like he has what they call questionable assumptions. There's like seven of them. And, he talks about, first of all, he has a huge table of every single cancer, basically, human cancer. And it's basically like, hey, here's the reference of every paper that has shown mitochondrial damage in these particular cancers. And it's like, okay, well, there's no way out of that one. Then they also talk about these lipid droplets being found in all these cancer cells. And that was believed, at least at one point, that, oh, well, they're just saving up the lipids so that they can use it later for beta oxidation. But that's not the case because basically, if the fat was to be able to be broken down in beta oxidation and actually burned into the mitochondria, it would produce so much reactive oxygen species and the environment that cancer is that it would terminally damage the cancer. So that I think I don't know if I can agree with that one, man. I mean, I know there's a lot of papers out there that muddy the waters, especially with titles and clickbait titles, man, that freak people out. People do videos on them that I don't think are as thorough, and they freak out my audience. I know that for a fact. And what I'll also say that is if that's true, then I would say that even the self-suppression theory would be false, right, for prostate cancer because even this, I mean, it's based off the cell danger response, and they're saying within minutes we're going to from oxphos to Warburg, right? So I don't know if I can buy that. What I'll also say is that I think this is something that's a nuancy thing, but it's not an all or nothing, right? It's not like Oxphos, yes, Warburg, yes. It's not that. It's if you look at the
Yeah,
graphics
there's a
that Seafried
nuance.
put out, it is a gradual decline. And I personally believe the more differentiated the cancer, the earlier the cancer, probably being realistic is probably a harder disease to treat than a metastatic, end stage disease because it's clear in the literature, whether it be Seafried or not, like the more aggressive and more malignant and more, metastatic the cancer is, the more the substrate level phosphorylation of glucose and glutamine is apparent as being the dominant paradigm. And it says in that paper, it also says that the more aggressive the prostate cancer, or if CLL gets converted into like a lymphoma, a more aggressive form, like it will then switch metabolically, right? So I do think that there's probably some degree of oxphos going on if it's allowed, because even if the mitochondrial structure is enough for some degree of oxphos to go down, it's not enough to survive, right? So that's not what Seafried is basically saying, that in the absence of glucose and glutamine, no cancer can survive on the amount of oxphos made at whatever level after transformation happens. It gradually goes down as time goes on. And the other thing that we're missing, that maybe both you and I haven't talked about at all, is the fact that these tumors are not having the same amount of oxygen than the rest of the tumors do, right? So how can you have, they talk about the cancer stem cells specifically about this. It's annoying. They'll say, oh, these cancer stem cells or these tumor initiating cells, they are in the most hypoxic, poorly vascularized regions of the cell. When you look at what that means in terms of like amount of oxygen, we're talking minuscule amounts of oxygen. But then they'll, in the same sentence, say, well, they have more oxygen consumption than even their normal counterparts. How does that even make any sense? You know what I mean? So there's just a lot, I think, to unpack there. I'm sorry I went on a little bit of a rant, but I
No,
think
that's
that is I can't accept that, from what I've seen.
no. Well, I love these rants. That's the whole point. So, yeah, I totally agree. It can be really difficult to determine the truth from the medical literature
It is
because there
hard,
are
man.
a lot of you got to look at where the study, who's paid for the studies, and where's the independent scientists and all that sort of stuff as well. Um, So yeah, it is very difficult, and I'm interested in truth. So I'm open to the concept that my theory could be wrong and the metabolic theory could be right. So I love these sort of interplays because we can try and flesh out a few details. The thing for me is that through the process, uh, as Professor Seyfried describes a lot of the metabolic theory components and how they fit with the disease. Um, he's absolutely right. He can explain a lot of things through this process of mitochondria becoming defective or oxphos becoming defective. However, what I'm highlighting is that um, there is this, you can literally explain virtually everything being explained in a different, through this different mechanism that I'm proposing. So, for instance, just there you mentioned lipid droplets. Uh, so, lipid droplet formation could be a sign, as Professor Seyfried states, that oxphos is inoperable and fatty acid oxidation cannot occur. So, therefore, lipid droplets are accumulating. However, lipid droplet accumulation isn't just a feature of that process, it's also a feature of infection. So, this is a feature, again, which ties into the cell danger response model, where mitochondria are intentionally suppressing the use of oxygen for ATP in order to generate free radicals. Um, Lipid droplet formation um, is generated during infection in order to protect the cell itself from the free radicals that could damage the polyunsaturated fatty acids that are present. So it encapsulates those because they're highly reactive.
Hmm.
So it's a process of defense, but also the lipid droplets themselves are immunologic. There's a feature that I was looking at a little bit earlier today, but there's a feature associated with them that they deal with the process in the body of aiding mitochondria and cells um, combat pathogens. So there's a very good reason associated with pathogen infection as to why the lipid droplets would be forming.
Hmm.
So you could explain it in two separate ways. So it's not a case of one or the other. And I'm
I
not
agree.
saying my way's right. I'm
I
saying,
agree with that. I
you
think there
know,
could be more than one explanation for sure. And I'll just
um,
preface, I mean, just so it's out there. I don't think anybody on earth has it all right, including Dr.
oh
Seyfried.
no,
But I,
yeah,
I, I do think that that's also the dominant paradigm that I base a lot of my, understanding on. But again, I don't think he has it all right because I think if somebody had it all right, there wouldn't be cancer. I mean, it would be eradicated. Like it would be it. So fully agree. There could be another explanation for the lipid droplets, but that is at least some piece of information that he gives as pushback to the fact that lipids are burned or oxidized as fuel. And I think that the number, structure, and function of mitochondria explain that, but also something that is talked about as much is the hypoxia
yeah,
issue. Because if there's no oxygen, there's no way for oxphos to actually functionally happen and couple to water, right?
yeah. There's so many different little avenues I'd want to go down. Um, One of the contentions I would mention is that oncocytomas are benign tumors, but they feature effectively defective oxphos, but they don't. Well, it's very rare that they turn into metastatic cancers. They are slow-growing benign tumors. So in that sense, you've got this switch to the Warburg effect because oxphos is defective, which would be right down at the bottom of that scale of Professor Seyfried's of what needs to occur for cancer to form. However, it doesn't develop. Um, these are benign tumors. And then going back to prostate cancer. So whether it's not whether this, the studies that I've looked at are in contention, like you say, you can't, no one particular study is necessarily as efficient at explaining the truth as any other study. Um, what we see from what I can gather from medical literature is that we essentially have the Warburg effect in healthy prostate tissue. And the reason why, I mean, I've provided a recent explanation for
Hmm.
prostate cancer, which ties perfectly in with my theory, um, to show that it's induced. And that is so the whole difference between the reason why you've got this metabolic abnormality compared to most other cancers in prostate cancer is because the initial metabolic profile is very different. So it's highly glycolytic, isn't it, initially? Um, the TCA cycle is truncated at citrate in order to produce the seminal fluid that's required for reproduction. And that means that mitochondria are shut off. But also fatty acid oxidation is upregulated in the sense that it seems to be that the fatty acids are being converted into the citrate where the glycolysis is being used to generate the ATP for the energy for the cell to function. So we've got high levels of zinc entering into the cell because that is what allows the truncation of the TCA cycle to occur. And then with prostate cancer, that's healthy tissue. So in prostate cancer, we see a shift where zinc is sequestered, drops down dramatically, and then we have a drop in uh, lactic. acid production, glycolysis production, and uh, OXPHOS is upregulated, or reactive oxygen species at the very least is upregulated. But we have this drop in glycolysis as well, from what I can tell from the papers. Um, and that process appears to be uh, the cell is now fully utilizing the TCA cycle. That ties into the cell danger response model and my suppression concept, where mitochondria can now use the oxygen or the low levels of oxygen that are present to combat the pathogen. Um, so half of the electron transport chain can be utilized through fatty acid oxidation for production of ATP energy, because it's citrate, like you said, is being exported within the tissue, and that's the precursor to fatty acid synthesis.
Hmm.
So you already have fatty acid oxidation upregulated anyway in that tissue, and you now have a lipid-rich environment. So there's no need to utilize glycolysis. Um, but the reason why this shift occurs, this drop in zinc occurs, and it's zinc, I think, is the pivotal mechanism that we need to look at to understand what's going on. And that comes back to the fact that zinc is required by pathogens, and the body knows this, and it intentionally suppresses zinc. So in any infection-based study you look at, zinc is sequestered. Now, this happens with iron, iron of inflammation. The reason the body sequesters iron away during acute inflammation is because it recognizes that iron is like rocket fuel to pathogens. So that's one of the main reasons it does that. So it's using the same process with zinc. So zinc is high in the prostate, and if pathogens get access to zinc, it makes them more virulent. So the response to an infection is to immediately drop the zinc down, so that zinc is no longer, uh, so pathogens no longer have access to the zinc. That then allows the TCA cycle to fully function, which then gives power back to mitochondria, essentially to utilize the oxygen to combat the pathogen, as per Robert Navio's model. And as well as that, because the pathogen is primarily glucose dominant, it's its primary fuel source. We also know that with what pathogens do, they outcompete immune cells for glucose. It's a strategy to defeat both the immune cell and the immune cell to defeat the pathogen. So I would also argue, because fatty acid oxidation is already present and available and being used, the cell also possibly reduces glycolysis for the purpose of reducing the amount of glucose that is available for the pathogen, as well as an anti-pathogen process, or simply because it's not needed, because there's enough oxygen to facilitate fatty acid oxidation at that level. And then that would explain later on, as the tissue grows, um, we have a reduction in the amount of oxygen availability occurring, so greater hypoxia, and then that forces the switch to glycolysis, which is why then the Warburg effect occurs later on in advanced pancreatic cancer. And then that enables the pathogen that's present to get access to the glucose that really drives the pathogen itself. Now, one key feature of my theory I do want to point out at this point is that you don't need every cell in the body to be infected by a pathogen to cause this switch in the Warburg effect, the anti-infection response. Because what we find is that you just need a small biomass infection in a number of cells to elicit a communal response in hundreds, if not thousands, of cells in the periphery, where they also switch to the Warburg effect, even though they're not
Because
infected.
of the local inflammation that's caused there and the
Because
cytokines that are there. Is that what the reason
because
is?
there's it's partly to induce inflammation, but it's as a communal response to infection. So, for instance, if a cell gets infected by a cell, sorry, if a cell gets infected by a pathogen, it's not that one cell that instigates inflammation. It's a number of the cells around it. They work communally together within a certain periphery. So, what you'll find within the tumor, you won't have an infection in every single cell of the tumor. You'll have a very small infection that elicits a huge response in hundreds, if not thousands, of cells. They'll trigger the Warburg effect in uh, in response to the infection, in order to help eliminate it, to eliminate the glucose in the local environment,
Hmm.
to stiffen the membranes of the cells so that the infection cannot um, uh, diffuse into other cells. So you're creating a hostile environment to contain the pathogen and help eliminate it and alert the immune system to the problem. So that kind of explains why, as Ravid Straussman and colleagues have highlighted, that you have intracellular pathogens within the macrophages within the tumor, but also sporadically within the tumor mass. Now, this, I would argue, is the reason why the immune system has difficulty targeting the tumor, because what you're asking the tumor to do effectively is, say, if there's 500 non-infected cells triggering the Warburg effect in response to the singular cell that has been infected, what you're asking the immune system to do is target itself. Cells that are actually doing what they're programmed to do, switch the Warburg effect to help eliminate the pathogen that's present. And the immune system's not going to do that. And this might also explain why we've got an issue with immunotherapy. You're generating an autoimmune disease in some cases. Obviously, it can be beneficial in others, but predominantly, as I think Seafried points out, that 20% of people have a beneficial response, 60% don't, and another further 20% have hyperactive disease, and we get there's an issue with autoimmune diseases occurring. And I would argue that's because you're trying to force the immune system to target largely uh, the cells in the tumor that are still self cells. They're not actually tumor cells in the sense that they've 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 changed or moved over to the dark side. They're being driven by this process of trying to eliminate the pathogen, and they're working in tandem with the infected cell or number of infected cells. So that's a key feature of my theory.
Got it. I think that for me, in some ways, and I'm maybe a little biased, maybe more than a little, um, I think it's hard for me to wrap my head around the idea of having such an occult infection that that causes this process to happen, activates the cell death, cell danger responses and has all this inflammation associated with it. Yet, there's really no symptoms from a patient, I think it's hard for me to wrap my head around, I guess, because was that not a great intro to this 2.5 nearly hour marathon conversation that we had together? It was a great overview of his cell suppression theory, and I really, really had a great time filming this and speaking to Mark. I actually ended up speaking to him for another almost nearly hour after the 2.5 hour podcast was over. And I really think he's a bright young man, and I really want to help him, hone his theory to make it as good as it can be. And I hope you liked part one. We'll be dropping part two later this week. If you like these videos, please like, share, and subscribe. And until next time.